Now, when we look at other factors in the environment that are going to drive or hinder microbial growth, the second one we're going to talk about, gases. What gases are actually present in that environment? Turns out oxygen is actually quite damaging, as we're going to talk about on the next slide. But when you take into mind, keep in mind, oxygen is damaging. What we find is microbes, along with most living things on this planet, are going to fall into three general categories. Those that can use oxygen and detoxify it. Those who can neither use oxygen nor detoxify it. Those who don't want to use oxygen but can detoxify it. Now, what do we mean toxify? Toxic. Toxic here for oxygen means that it's going to be stripping hydrogens off of anything it comes into contact with. Whatever compound that oxygen is in, if it's, in need of more valence electrons, outer shell electrons, it's going to strip them. And basically, when it comes down to it from a biological standpoint, anytime people are discussing free electrons, things like that, they're talking about hydrogen. Hydrogen is nothing more than a proton and electron. So oxygen's going to strip hydrogen off of things. It's going to strip electrons off of anything it can. So what forms are we talking about here when it comes to really bad oxygen? Well, first there's the singlet oxygen, just O. Not O2, just O. Oxygen, singlet like that, well, it's going to start stripping hydrogen, it's going to start stripping electrons off of anything it comes into contact with. Membranes, proteins, DNA. The last one there, DNA, that's where it can be, has the potential for being the most damaging. You also have superoxide anions, O2 negative. These are going to start stripping protons and electrons off of anything they come into contact with, just like the singlet oxygen. Hydrogen peroxide, yes, the stuff from the brown bottle your mom used to pour on your boo-boos and it would bubble. H2O2 breaks down into water and superoxide anions. And then finally, hydroxyl radicals, OH. The hydroxyl radicals usually are on some organic molecule and they get cleaved off, or hydrogen peroxide or water gets broken down. So you have to be able to take care of these. We, mammals, pretty much most mammals, animals, are going to have the three enzymes in red. How do I know they're enzymes? Rule of thumb, anything that ends in A's, ASE, means it's an enzyme. Whatever prefaces A's is usually either its target or its activity. Superoxide dismutase takes superoxide anions and some free electrons, some hydrogens, makes regular oxygen, O2, which is good, and then hydrogen peroxide, which has the potential to be bad. Well, hydrogen peroxide can be broken down in one of two ways. Peroxidase, which is going to strip off some hydrogens from NADH, and it's going to take peroxide and it's going to make water. Or catalase, which is going to take peroxide, a couple of peroxides, and it's going to make water and oxygen. Turns out when mommy poured the peroxide on your boo-boo and it bubbled, it's the catalase in some of the enzymes as they're breaking down the hydrogen peroxide. They were making water and gaseous oxygen, bubbles. Not all bacteria have these three abilities. So it's one of those things you can actually test for in a lab. Of the Streptococcus species, the Staphylococcus species, not all of them have, or the staph and the streps, not all of them have catalase activity. So you can take hydrogen peroxide, stir in some bacteria from a colony, and test whether or not it bubbles. If it bubbles, catalase positive. If it doesn't bubble, catalase negative. If it bubbles, Staphylococcus aureus or one of the other staph species. If it doesn't, it's not. Easy peasy. Not all bacteria are going to have these abilities. Those that don't, which was number 2 on the list from the last slide, then any of the oxygen radicals you see there, which also are referred to as free radicals, they can cause damage. Oxygen will be toxic. Where can you find these in your body? Large intestine, between your gum and your teeth and your mouth. You're going to find true microbes that cannot handle being in the presence of oxygen. And then that third category, well, those bacteria have one of or some of the 3 listed there, but their metabolism isn't based on oxygen. It doesn't need oxygen to be the last electron acceptor for, the electron transport chain for the making of ATP. So, based on who lives, who dies, who doesn't care when it comes to the presence of oxygen, 5 categories. Aerobes, like us, have to take in oxygen. Without oxygen, metabolism, ATP production shuts down. 2, microaerophiles, like oxygen, but not too much. They can't detoxify it fast enough so it becomes damaging. So they want really, 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 really minute concentrations. These are microbes you're going to find like armpits, groin, in your mouth also, where there's oxygen, but just not a lot. Facultative anaerobes. These are going to be anaerobes that, oxygen's there, they can use it. If it's not, oh well. They have other mechanisms. Think E. coli, Staphylococcus aureus. Anaerobes, no oxygen. Keep oxygen away. They do not have the ability to detoxify it, and it's deadly to them. And then finally, aerotolerant anaerobes. They don't use it, and if it's there, they'll just ignore it. They have no metabolic necessity for oxygen like we do. So when you look at the different categories here from table 6.5, keep this in mind. This is also going to impact Koch's postulates. That postulate number 2, can you grow it in the lab? We have to take this into account, are we giving the nutrients, and also how much oxygen are we giving this thing? Give it too much? Are we killing it? Don't give it enough? Are we starving it? Just having it there, can that be deadly? We could test this in the lab. You see, whichever side it's on, the tubes of broth. Tube 1, you can see that there's not very much growth in it, bacterial-wise. You don't see a lot of it clumped together like you do in tubes 2 through 4, because it's all right there up at the top. This tells us that these microbes are anaerobic. They have to be up at the top of the water column, because up at the top is where there's the maximum amount of oxygen. As you go down through the tube, down through the liquid, you get less and less oxygen. So that down at the bottom, there's almost no oxygen. Tube 2. Notice it's the top half. These are going to, but if you look, you can just barely make out that up at the top, not so much. These are going to be the aerotolerant, or the microaerophiles. They like it, but not too much. Number 3, ubiquitous, all the way through. These are going to be the facultative anaerobes. They don't care if oxygen's there. They can do without it. And then 4, nothing growing at the top. It's all from almost the midpoint down. anaerobes. These are the ones that do not want to be where there's oxygen present. We also find that there are microbes that actually need CO2. They need higher than normal, higher than atmospheric CO2 levels. Neisseria gonorrhea, Streptococcus pneumoniae. They need higher CO2 levels, and we refer to these as capnophiles. Oxygen, yes, but high CO2 also. Now let's change for the last bullet point here. Just like we talked about with temperature, just like we talked about here with oxygen, pH is also going to be a too hot, too cold, just right Goldilocks thing. Too acidic, too basic, just right. Microorganisms are going to have certain ranges where they want to grow.